Hi, today I will be talking about the limiting factors of photosynthesis and chromatography, and these are part of 2.9 molecular biology under photosynthesis, so it's both standard level and higher level IB Bio need to learn this. So first I'll be talking about the limiting factors involved in photosynthesis. There are three, those being light intensity, carbon dioxide concentration, and temperature. So I'll first start off with light intensity. So at low light intensities, the rate of photolysis, and therefore the production of oxygen, is limited by the amount of light absorbed. Photolysis essentially splits water into oxygen and other products. And as the light energy is used for the production of ATP and high energy electrons, which are both needed for the conversion of carbon dioxide into glucose, low light intensities limit the production of this sugar, so glucose, and other useful substances. If there was to be a high light intensity and photosynthesis is being limited, then it's a different limiting factor. As you can see in this graph, really only low light intensities will affect the rate of reaction, and after a certain point, the rate of reaction plateaus off and stays the same. Unless a plant is very heavily shaded or the sun is rising or setting, light intensity is not usually the limiting factor. Essentially, it limits photolysis, and therefore also limits the production of oxygen. So the second limiting factor is the concentration of carbon dioxide. And below a certain concentration, it's around 0.01%, the enzyme used to fix carbon dioxide, which is called rubisco, is not effective, and in many plants there's no net photosynthesis. As the concentration of carbon dioxide increases, the rate of successful collisions between the carbon dioxide molecules and the active site of the enzyme, so rubisco, is going to increase. At low concentrations of carbon dioxide, ATP and the electrons, which are very high in energy, are not used as rapidly as they are produced, which will then restrict further photolysis and therefore oxygen production. And so if there are, as you can see in the graph, again similar to the light intensity graph, the rate of reaction will plateau around here, and so really only a low carbon dioxide concentration will cause photosynthesis to be limited. So if there is a very high carbon dioxide concentration in the atmosphere, currently it's 0.04%, if that concentration has been reached, then and photosynthesis is still being limited, then some other factor is limiting. The final limiting factor is temperature, which, as you can see in the graph is clearly differently shaped as opposed to light intensity and carbon dioxide concentration. So at low temperatures, all of the enzymes that catalyze the conversion of carbon dioxide into carbohydrates eventually work slowly, and below a certain temperature there is little or no photosynthesis in many plants. This temperature is around like 5 degrees Celsius. At temperatures above 30 degrees Celsius, rubisco, which is used to fix carbon dioxide, is decreasingly effective, even though it has not yet been denatured. So if temperature continues to increase past this 30 degree Celsius threshold, enzymes, so including rubisco, but other enzymes used in photosynthesis, will eventually start to denature. Temperature is a limiting factor at both low and high temperatures, where the low rate use of ATP and the high energy electrons will restrict further photolysis and again therefore oxygen production. So at intermediate temperatures, so within this range here, if photosynthesis is being limited, then it has to be one of the other two limiting factors. That's pretty much it for limiting factors. So moving on to chromatography. Chromatography is an experimental technique where mixtures made up of pigments can be separated. So obviously in plants you have pigments within plants, including chlorophylls, xanthophylls, and carotenes, carotenes. However, chromatography can also be done on ink, for example, as different inks are made up of different pigments, and this will separate it. So a mixture is dissolved in a fluid and then passed through a static material. And there are two techniques for separating photosynthetic pigments. The first one is paper chromatography, which uses cellulose, so paper, as the stationary bed and the static material that the mixture is traveling upwards. So this paper here. And the second type of technique is called thin layer chromatography, which uses a very thin layer of adsorbent, which runs a lot faster than paper chromatography and has better separation just because of the material that is being used. Because the different components of the mixture, the different pigments, travel at different speeds up the paper or the adsorbent, they will separate, as you can see in this image here. So the mixture started down here, and as the solvent traveled upwards, the different pigments start to separate. From this, after the pigments have finished separating, an RF value can be calculated for each individual pigment, which is the distance that the pigment travels over the distance that the solvent travels. For every single pigment, the distance that the solvent travels will be the same because the solvent will travel up to a certain point. So for example, in the diagram, the solvent will travel up to here. However, the distance the pigment travels obviously depends on the pigment. So for example, for xanthophyll, which is the yellow pigment here, you would do the distance, so let's say that's like 5 centimeters over the distance that the solvent travels, which is, let's say, 10 centimeters. And so the RF value would be 1/2, so 0.5. And obviously this will be different for every pigment. That's pretty much it for chromatography.